Hello, IB Biology students. This is your next installment of today's video lecture notes. Today we're going to be looking at Chapter 5, Section 3, Classification and Biodiversity. Remember to take notes and write down any questions that you may have so that we can review them in class when we are going over the key points. All right, the first thing that we need to remember is the way that we name organisms. So we use what's called the binomial nomenclature, which binomial means two named, and nomenclature or nomenclature means naming system. Excuse me. This system was developed by Linnaeus, who was a Swedish botanist and taxonomist. We're still using it today, and it's the basis of classification for naming all new species. This system works for allowing all of our previously discovered organisms and allows for the introduction of new organisms as we continue to discover them. So the way that this works is your two names are your genus and your species. So for example, we humans, our binomial nomenclature name is Homo sapien. So the first Homo is our genus, and the second sapiens is our species. We always write in italics if we're typing it, or we underline it if we're handwriting it. The first word, the genus, is always capitalized with first letter, and the species is always lowercase. So for example, we have the Homo sapiens, but then we also have the Escherichia coli or E. coli, and you can always also abbreviate the genus name, such as this S. cerevisiae. If you have, and this is only done if you've already written the full name out in your text. The binomial nomenclature, the two names, the genus and the species, are based on the physical traits of the species, and most of the groups that Linnaeus picked or named are still accurate to this day. He was very ahead of his time. So I have a quick little video, 3-minute video, to show you about Linnaeus.
Carl Linnaeus was born in the southern Swedish province of Småland in 1707 and is considered to be one of the most influential scientists of his time. His life's work was to develop and refine a way to group life on Earth, or to classify it. So straightforward was his system, it is still used by scientists and understood by people across the world today. His father, Niels Ingmarsson Linnaeus, was both an avid gardener and a Lutheran pastor, and Carl showed a deep love of plants and a fascination with their names from a very early age. While studying medicine at both the universities of Lund and the University of Uppsala in Sweden, he devoted a large amount of his time to the study of plants, and in 1732 he mounted a botanical expedition to Lapland. Over 5 months he travelled some 3,000 miles, collecting biological specimens and taking notes. In 1734 he mounted another expedition to central Sweden.
He was an empiricist. He didn't believe things that he hadn't seen with his own eyes. He was very reluctant to take on trust something that somebody had written about.
In 1735 Linnaeus moved to the Netherlands, and it was in that same year that he published his most well-known work, the Systema Naturae, or the System of Nature. In it he outlined a new system for the naming of all living things.
There are two main things that Carl Linnaeus tends to be known for. The one is as far as his classification systems, which at the time were really rather revolutionary, particularly in the plant kingdom, rather less so amongst the animals. And the second thing is his introduction of a new naming system using a genus name and a species name, the so-called binomial system. Now, of the two, it's the latter, it's the naming system that is his primary legacy.
This item is the first edition of Systema Naturae, the one where Linnaeus actually outlines his sexual system for classifying plants, basically by the number of stamens and the arrangement of the reproductive organs. So this is the first time it's appeared in print, and so that's why it's important. You can see it's very large, and I tend to explain it as being the 18th century version of an Excel spreadsheet, because you can see it's very table-like. By the time the 10th edition appeared in 1758, this had shrunk from this really large size to a much more sort of portable thing, but it had gone from only 11 pages to 2 fat volumes.
Returning to Sweden in 1738, he practiced medicine, specialising in the treatment of syphilis, and lectured in Stockholm before being awarded a professorship at Uppsala University in 1741. Throughout his life, Linnaeus showed an interest in nature and the systems which govern it. He wrote not just about classification, but also about ecology, how organisms interact with their environment. He explored food chains and even defined the concept of race, dividing humans into 4 groups: Americanus, Asiaticus, Africanus, and Europeanus. Forced to retire from teaching in 1774 by a stroke, Linnaeus suffered a further stroke and eventually died in 1778. Linnaeus was driven by a lust for all nature and the desire to understand and classify it. His legacy remains and is used by the many dedicated scientists today who are driven by that same desire.
All right, so with this idea of a binomial nomenclature, we obviously, as we just talked about, use the species and the genus, and those are 2 levels of taxonomy or levels of classification, taxa. So taxonomy itself is the practice of science, is the practice and science of classification. There are 7 levels of classification, with the largest being the domain, and then working our way down to the species through domain, kingdom, phylum, class, order, family, genus, species. This system allows us to group organisms while also allowing us to assign unique species names, and it's very helpful in identifying and naming new species. So as you're working from the largest to the smallest, as you move down, the organisms within this area or the next level are going to share more characteristics with the organisms that are in there than the one that they were in above. So as we move down, once we get to the genus, all the organisms that are within the genus level of taxa are going to have more characteristics that are similar to each other than, say, all the organisms that are in the order group. All right, so what exactly is a species? We are going to get more on in this later, but in general, a species is a group of organisms which can interbreed and produce fertile offspring, and that is a very, very important part of the definition. They are the smallest taxonomic group, although we do actually recognize what we call a subspecies for many organisms. Subspecies might potentially interbreed if a barrier or other challenge was removed, and the most common separation for subspecies is distance. So here you have 2 subspecies of a white peacock butterfly, and so they do not interbreed and produce fertile offspring, but it is because of their location that they do not. So we call them subspecies. Jamaica and Puerto Rico, of course, being separated by a large body of water, and if that large body of water were to go away, if these 2 islands were to combine, that these peacock butterflies would be able to reproduce and produce fertile offspring. All right, so let's talk about the differences in the 3 domains. So we do have 3 different domains. We have the bacteria, the archaea, and the eukarya, and the DNA is different. So DNA in bacteria does not have what we call proteins or histones associated with it. It's just a single coil of DNA itself. Archaea do have proteins that are similar to histones, but they're not histones. Those proteins are bound to the DNA to help it condense and pack. And then eukarya does have the histones associated with the DNA, and those histones are for condensing and packing the DNA, especially in times of division. Introns, remember from your early biology days, are the sections of the DNA which are not for coding for proteins. So they're extra sections of DNA. They could be used in regulatory processes or in determining which sections of the DNA do need to get transcribed and translated. Bacteria do not have introns, or if they do, they're very, very rare. Archaea have some introns for some genes, but again, not very common. But eukarya have many, many, and a frequent presence of introns. Bacteria, when they have cell walls, that cell wall is made of a structure called peptidoglycan. Archaea have a cell wall, but it's not made of peptidoglycan. So you can actually stain for this peptidoglycan and determine whether you have an archaea bacteria or a eubacteria because of the structure of the cell wall. Eukarya does have a cell wall. It's not made of peptidoglycan, but it's also not always present. The most common cell wall, of course, in eukarya is our plant cell wall, and that's made of cellulose. The cell membrane is similar in that they all have what we call glycerol ester lipids, and they all have unbranched forms. But the bacteria and the eukarya cell membranes are similar. They have unbranched side chains of what we call a D-form glycerol. So it's just a different form. There's two different forms of glycerol, an L form and a D form. And both bacteria and eukarya have the D form. But archaea use the L form of glycerol. All right, before we go into too much more detail into the next section, I want to review what a dichotomous key is. This is a tool that allows users to determine the identity of a certain organism without knowing what it is. Dichotomous means divided into two parts. Therefore, dichotomous keys are always given two choices in each step. So if you look, they can be in they can be in several different forms, but your two most common are in a branching form or in a table form. So in this branching form, you have, does the organism have feathers? So you would look at your organism in front of you. Yes, it has feathers, or no, it has feathers. So let's say that our organism does have feathers. And then your next question would be, does it swim or does it not swim? So say ours does not swim. Then according to this, we would have a hen that we would be looking at. In the table form, it's very similar. You still have a question. You have one question with two stems to it. The organism has a backbone or does not have a backbone. And then instead of using a bracket system, you would pick your answer. So say we have a backbone, and now we are going to go to question 2. And now we look at these stems here. So the organism has wings or does not have wings. So if he has wings, we would go to question 6. So we skip down. Does the organism have feathers or does it have hair? Our organism has feathers, so we are looking at a bird. So just two different ways of looking at dichotomous keys. We will in class be making dichotomous keys and then utilizing our peers' dichotomous keys to see if we can figure out what the organisms are. All right, so that was really important because we can use that branching also to look at our breakdown of organisms as well. So in the kingdom Plantae, so this is within the domain Eukarya, we have two main different structures of plants. We have what we call non-vascular plants or vascular plants. Non-vascular plants are very small. They have no conductive tissue. So your most common example that you're going to think about is mosses. Vascular plants do have conductive tissues. We call those conductive tissues xylem and phloem. And then those vascular plants break into two different types, spermatophytes or follicophytes. And then the spermatophytes break into angiospermophytes or gymnospermophytes. And then angiospermophytes break into monocotyledons or dicotyledons. And we're going to look at these in more detail in a table on the next slide. Next 2 slides. So here are some pictures of the ones that we need to be responsible for. So we need to be responsible for Bryophytus, Follicophytus, Coniferophytus, and Angiospermophytus. All right, so looking just at those 4, let's look at each column one at a time. So vegetative organs. Bryophytus do not have what we call true roots. Instead, they have rhizoids. And then all of the rest do have roots, stems, and leaves. So they do have all three vegetative organs. In the vascular tissue, as I said earlier, bryophytes have no xylem and phloem. So xylem and phloem are the vascular tissues that allow for food and water to transport up through the plant, whereas the follicophytes, the coniferophytes, and the angiospermophytes all do have that xylem and phloem. Cambium. Cambium is the inner bark. It's not the outer bark, and you can see here in this picture down here, we have our outer bark, and then we have our cambium, and then you get to the wood, and then the pith in the middle. So both Bryophyta and Follicophyta, Bryophyta, remember, is moss. Follicophyta are ferns, so neither of those have the cambium. Coniferophytes are cone-bearing plants, and Angiospermophytes are flower-bearing plants, so they both do have a cambium. This is the same thing that is same rule of thumb for pollen. Neither Bryophyta or Follicophyta have pollen. They reproduce by spores. But Coniferophyta does have pollen. Pollen is actually produced in the male cones of the trees, whereas in Angiospermophytes, they're produced by the anthers or the flowers. So that is the male sex organ, and then the ovules being the female sex organ. Again, the Bryophytes and Follicophytes reproduce by spores, so they do not have ovules. And the Coniferophytes and the Angiospermophytes do have ovules. So Coniferophytes, most people don't realize there are male cones and female cones. So in the male cones, there's pollen, and in the female cones, there's ovules. In the Angiospermophytes, that is where they have flowers, and there are two different places in the flower where the pollen and the ovules are. So they could be in the same flower itself and self-pollinate. So the pollen is within the anthers, and those are the right, so the pollen is produced in the anthers. So here are the anthers within this flower molecule, and then the ovaries are within the flower itself, within the ovule and the stigma of the pistil. So again, Bryophytes and Follicophytes, moss and ferns, do not have seeds either, still reproducing by spores. Coniferophytes do produce and disperse seeds, and as well as Angiospermophytes. And then our last column, fruits, neither Bryophytes, Follicophytes, or Coniferophytes produce fruits, but Angiospermophytes do. The fruits are produced for seed dispersal. So these are could be fruits that we eat or fruits that are poisonous. So berries, apples, any of those are going to be the seeds, or a covering for the seeds so that those can be dispersed throughout the area by other organisms. All right, within the kingdom Animalia, you have lots of invertebrates and vertebrates. That's one of your first main divisions. And these are heterotrophs. They are animal cells. We have 6 invertebrates that the IB curriculum would like us to be aware of and responsible for. And I'm going to go through these in a table format, just like I did for the plants. Please do remember that I will have samples to look at in the classroom, and we will be reinforcing this knowledge when we look at those organisms, okay? It is a bit overwhelming. All right, so our 6 organisms that we need to be responsible for: Porifera, Cnidaria, Platyistomys, Annelida, Mollusca, and Arthropoda. All right, so the first thing, again, when you're starting in this table, the most simple organisms are at the top and the most complicated are at the bottom. So when we look at whether there's a mouth or an anus, the Porifera, that's a sponge, has no mouth or anus. The Cnidaria and the Platyistomys do have a mouth, but they have no anus. And then the Mollusca, the Annelida, and the Arthropoda do have both mouth and anus. So along that same lines of going from less complicated to more complicated, we're going to look at the symmetry next. So Porifera has no symmetry. Cnidaria has a radial symmetry. A common example of a cnidaria is a jellyfish. So if you think about that, you can see that radial symmetry that they would have. Platyisthmes is going to have a bilateral symmetry. A common example of these are platyisthmes. Flatworms. All right, so a common example of a platyisthmes is going to be a flatworm. A mollusca has and all the rest have bilateral symmetry. A mollusca is like a slug. An annelida is a segmented worm, and an arthropoda would be an exoskeleton-based insect. All right, so Porifera, as far as the skeleton is concerned, they don't have any type of external skeleton. The only internal skeleton they have is they have these things called internal spicules, and they're like skeletal needles, but it is not a full skeleton itself. Cnidaria are a soft organism. There are some cnidaria, corals are cnidaria that secrete calcium carbonate, so they are hard on the outside, but then the actual organism itself is a soft and has no skeleton. Platyisthmes are soft and have no skeleton. Mollusca always have a shell made of calcium carbonate. Annelida have an internal cavity with fluid that will fill under pressure. And arthropoda has an external skeleton made of plates called of a molecule called chitin. All right, so some other external features to be aware of. Porifera, again, being sponges, have many pores because they are filter feeders, and they have variations in shape. That's why they have no symmetry. Cnidaria typically have tentacles that are arranged in rings around their mouth, and many have stinging cells. Remember, our example here is jellyfish. Platyisthmes have a flat or thin bodies that are in the shape of a ribbon. They have no blood system and no system for gas exchange. A lot of your tapeworms or parasitic worms are platyisthmes. Mollusca has what we call a mantle, which is a fold in the body wall, and that mantle is actually what secretes the calcium carbonate to form a shell. They also have a hard, rasping radula for feeding. A snail would be a great example of a mollusca. Annelida are organisms that have bodies made of very many ring-shaped segments, so we call them segmented organisms. They often have bristles, but they can also have blood vessels that are visible. So earthworms would be an annelida, and then centipedes and millipedes would be annelida. And then our last group is our arthropoda. These are segmented bodies with legs and segmented bodies and legs with joints between the sections. So grasshoppers are a really good go-to example for arthropoda. All right, so now we need to look at some vertebrates. So before we were looking at invertebrates, here is an evolutionary tree of some of our vertebrate animals. And notice that we have, as we go through, we have a lot of swimming things. And then here we have where four legs evolve. So just these three have four legs, although a couple of these have leg-like organisms. Leg-like organs. All right, so we need to know 5 different phylums of vertebrate organisms, or 5 different organisms within the phylum Chordate. So we need to know about bony rayed fin fish, amphibians, reptiles, birds, and mammals. And we need to know a few different pieces of information about them. So starting off, our outer surface, our bony ray fin fish have scales. Scales are actually bony plates in the skin. Amphibians have a very soft, moist skin that is permeable to water and gases. Reptiles have an impermeable skin that is covered in scales of a protein called keratin. Birds have a skin covered with feathers, and those feathers have keratin in them as well. And then mammals have mammal skin has follicles with hair that is also made of keratin. So notice that once we get into the impermeable skin, we have some type of keratin covering the skin as well. In gas exchange, our bony ray-finned fish have gills. And then as we move through our gas exchange, we're going to get more and more complicated. Amphibians have simple lungs with very small folds, whereas reptiles have lungs with more folding or extensive folding. Birds have developed what we call parabronchial tubes and then air sacs. So they have very simple tube system with air sacs at the end. We as mammals, or part of the mammal group, have lungs with what's called alveoli, ribs, and a diaphragm. So we have many branches within our lungs. All right, so we've got a couple different pieces of information with appendages. Bony ray-finned fish have no limbs, but they do have fins that are supported by rays. All the rest of our organisms here are what we call tetrapods. So remember, tetrapods are 4 legs, and each of those legs have what we call a pentadactyl limb system. So that's the one bone, many bone, little bones, and then digits. Amphibians, though, only get the 4 legs when they're an adult. Reptiles, most species have 4 legs. Birds have 2 legs but 2 wings, even though they are a tetrapod. And then mammals have 4 legs, but can also have 2 legs or 2 wings or 2 arms. So depending on the mammal we're looking at. All right, for our development, our bony ray-finned fish remain in the water for their life cycle. Amphibians are in the water for their larval stage, but are on land for their adult stage. Reptiles, the female will lay eggs, but the egg will have a soft shell. And then birds, the female will lay eggs with a hard shell. And then mammals give birth to live young and are fed with milk. Miscellaneous information, bony ray-finned fish have a swim bladder, and that swim bladder helps them with buoyancy. Amphibians, their eggs are coated in a protective jelly. Reptiles have teeth, which is interesting. Most students don't realize that. Birds have a beak, but no teeth. And mammals have teeth of different types, and inside the teeth is actually a living core. And then the last piece of information is whether we maintain our body temperature or not. So bony ray-finned fish, amphibians, and reptiles do not maintain a body temperature internally, and birds and mammals do maintain a constant body temperature. All right, that was a lot of information that you're responsible for. Please remember with those 3 charts of information, we are going to go over them. We're going to go over the plants, the invertebrates, and the vertebrate chordates. Okay, so please don't be too overwhelmed, and I'll see you in class.